Good. Good morning. And when you pop in, just type here in the chat for attendance. Thank you.
Good morning.
Good morning.
Y'all, I've been on a live TV kick, and it's so trippy to watch where I live be on it.
I don't know if that was for me. I couldn't hear well.
It was just a generalized speaking.
Oh, sorry. It's kind of, let me try to adjust my, it's a little loud. Maybe it's get some feedback. Maybe type if you have, it was still a little garbled, but maybe we're okay. Sorry. Okay, we're getting in the chat here. Okay, so where I live on on PD.
There's a show. They follow the cops around. Lawrence County or the Lawrence Township does it, and it's so weird to be like, I live there. Why is there a cop chase? And I didn't hear about that.
Oh,
I live there.
I got you. Okay. Yeah.
Lawrence is the worst, man.
Me and Emily are just like, Lawrence is a hot mess. I've seen them filming multiple times.
Oh, I've been in one. I'm in the background of one at Speedway.
Oh, that's funny.
There's this one guy that was cracking. He was on drugs or something, but he was talking about like Peeps and stuff. And he was just going off to the cops about Peeps. In reality, he was getting kicked out of Speedway. But they were trying to like avoid the whole conversation. He was stealing like all their liquor and stuff. But he was trying to say he was just trying to buy some Peeps. And then he was going on about his favorite flavor of Peeps, what color Peeps, how much Peeps have increased in price since he was a little kid.
Oh my gosh.
Like, Lord.
It's wild.
Interesting.
It was
Yeah.
like there was a fight at Walmart on one of the episodes, and I was like, of course.
Oh God.
Yeah.
I will not step foot in Walmart. I have to be seriously desperate for something to go into a Walmart.
Me too.
I can't do it. I just can't. Just typing. I'm clicking people off here as I will let some more people get on, and then Gonzalez, Keller, Carlo, Finley. Let me just trying to get ahead of the game. Oh, actually, I don't need to be recording this. Probably start recording again, and then let me see if I can do share my screen. Wait, now everything disappeared. Oh, because I'm stupid. Okay. There we go. As I just said, I'm getting better with Zoom, and I keep screwing things up here. Okay. Why is this not? There we go. Okay. If you're just getting in, just keep typing here in the chat. I mean, if you've already typed, you're fine. To let you guys, let me, where's everybody here? Oh, over here, shoot. Okay, I've muted everybody, but as always, you should be able to unmute if you have a question, or you can just type it. I just emailed you, and what I did is I tried to do, Zuber helped me yesterday. So what I did is I created the assignment in Teacheries. And so it's an assignment in Teacheries, and you should be able to upload. And I attached in this Zoom email that I just sent you, I attached the homework sheet. And so hopefully you can just type your questions on the answers on that, or I guess you could print it and handwrite it. And then you can upload it to Teacheries. So we'll see if that works. I did also, if you didn't read the email, the top line, there is one exercise in your lab book I want you to do, which is it's chapter 8 in your lab book, which should be the nervous system, but it's only exercise number 13, which is a list of terms. The vocab is not, even if the list looks long, it's not like it's massive definitions. And that's what I want you to do. I think I put to turn it in by 11:30 on Tuesday. And most of the answers should be in your nervous system chapter for the homework sheet. You should be able to find all of that in the chapter or in the chapter 8 lab book. So hopefully everybody got the homework, because we don't have school tomorrow. So you get a day off, get ready for Easter. So I'll just pick up where we left off if nobody has any questions. Okay, can you see the slides on the screen? I think you should be able to. Okay, good, thank you. Okay, so if I back up a few slides okay, my mouse, where did my mouse go? Oh, please tell me I did not run out of batteries. There we go. Sorry, I have no mouse. Can you hang? Yes, technical difficult. Wow, just when I thought I was doing okay, I have the light. Can you hang on 2 seconds? I think I have some batteries in the kitchen. Hold on. I may need to get my son. Hang on 2 seconds. Be right back. Hey, Gabe. Hey, Bobo. Okay. All right, let me see. Here, I have a mouse. Okay. Bobo, you want to say hi to everybody? Come here. Come say hi. He just woke up. You say hi? Big yawn. Technical difficulty. I thought I should have having. Okay, Michaela, if you get kicked out, we'll try and fix it. Maybe you can get some notes. I'll make sure. Like I said, I'm going to try this weekend, hopefully, to get these videos. You gonna leave me? I'm going to eat your breakfast. Okay. All right. Yeah, my internet just said my internet's unstable. So we'll try to get through as much of this as possible. Okay. So hang in there. When I was talking yesterday about this axon, And this is when you have a nerve fiber, you have multiple axons that are bundled together. They are surrounded by, there's something, most of the neurons or these axons are covered in something called myelin. Myelin sheath is actually made up of those glial cells. The glial cells are the supportive cells of the neuron, and they make like an insulation. So the myelin sheath, if you look on the picture, the nerve fiber is here. So this is multiple axons in that nerve fiber. And then you've got this myelin sheath, which is made up of the cell membranes of the glial cells. You've got oligodendrocytes, which is a nerve cell. And then you also have Schwann cells. All of those come together to create myelin sheath. And if you think about like an electrical wire, I think about movies and they talk about the green wire, the blue wire. Sean, you're good. You're all good. We just kind of, I had technical difficulties, so you're fine. But if you think about those wires in like the movies, that coating, that colored coating, if you peeled that off, there would be a copper wire underneath. You could think about the copper wire as the nerve fiber, and then the myelin sheath being the coating. What myelin does for the nerve is it helps the nerve send a faster signal. So a myelinated axon is going to send a signal faster than an unmyelinated. Now, most of the unmyelinated axons or fibers are found within the brain. Anything that is myelinated is going to be called white matter. If you talk about white matter versus gray matter is found in the brain, and that does not contain myelin, okay? But most of your peripheral nerves are coated with myelin. In the spinal cord, you have myelinated fibers. So think about myelin. It's like a coating or a protective insulation, helps send the signals faster. And that's what they're showing in this picture. So multiple oligodendrites and all of these Schwann cells are going to cover that whole axon. What you're going to notice is these little gaps. It almost, think about like links of sausage. You think about like you have sausage and then it kind of pinches, and then sausage and it pinches. If you look at this picture, okay, if you see all of those little indents, that whole thing is a Schwann cell. The indent is called the node of Ranvier. So the node of Ranvier is a gap in between those cells. Now, I'm going to talk about how a nerve sends a signal. The signal needs to jump that gap. So everywhere there's a gap, the signal is going to jump to the next cell, to the next cell. It goes kind of in an order. So myelinated axons all have this structure. Now, within, we talked about, obviously, the CNS, brain and spinal cord is on the axis. The peripheral is any part or component that extends off of the brain or the spinal cord. So the cranial nerves extend from the base of the neck near the cranium or the brain. The spinal nerves originate from the spinal cord, and you'll see the spinal nerves. Now, this was the important thing, and I kind of introduced this topic yesterday, but I was talking about afferent versus efferent fibers. Afferent signals come in to the brain. So when I detect a hot stove, that signal coming to my brain is afferent. It's coming in to the brain. And so typically afferent nerves are sensory nerves. So anytime you sense something, pressure, heat, cold, that is being sensed by an afferent or a sensory nerve. After it goes to the brain is going to integrate a response, and it's going to send us an appropriate signal on what to do. Because remember, I said the muscles are controlled by the nervous system. So the brain is always kind of processing and saying which muscles need to move. So efferent fibers carry impulses away from the CNS, and these are typically called motor nerves. Motor, movement, muscle. Motor, movement, muscle. The way I also think about efferent nerves is efferent nerves are affecting something. So when you touch the stove, the effect you're looking for is you move your hand, okay? You had to sense it, and then the brain sends an efferent signal and moves it. The other thing I think about is A comes before E in the alphabet. You have to have an afferent sensory signal before you effect a motor response. A versus E, afferent, efferent. Now, there are nerves that in the body that will have a mixed, you're going to have sensory and motor nerves. Most of the nerves in the peripheral nervous system, they have to do both jobs. So basically, any nerves that go to your arms and legs, if you think about it that way, they have to be able to send a signal, but then also transmit a signal to do something. So typically, the peripheral nervous system is all mixed nerves. But the main thing I want you to know is you have afferent is coming in, efferent is leaving. And that's what we're going to do. I noticed some people have bopped on. I know people maybe had issues. So if you've just joined, make sure you type that you're here in the chat so I can put you down for attendance. Okay, because it looks like a couple people just joined late. Okay, which is fine. As long as you join the chat, you get attendance. So make sure. Okay, Alicia. Yeah, sorry. Hang in there. Internet's wonky. Now, I talked about the CNS and the PNS, but there is also another kind of division of the nervous system that you have to think. There is the autonomic nervous system, and there's what's called the somatic nervous system. Think about soma means body, and you have conscious control over your skeletal muscles or your main body. So the somatic nervous system is under conscious or voluntary control. I can choose to sit in this chair. I can choose to walk into the kitchen to get batteries. I mean, my brain, I had conscious control. The important other side of this is the autonomic nervous system. Think of it as autonomic, automatic. I don't have to think about things that are controlled by the autonomic nervous system. They happen automatically. So this would be like digestive juices. If you eat a meal, automatically the minute the food hits the stomach, there's a secretion of digestive juices. It wasn't like, oh, maybe I should secrete some digestive juices. Okay. So movement of our GI tract or smooth muscles, expansion of our bladder, movement in the liver, movement in our esophagus, all of that happens automatically. So that's what I mean by autonomic, okay? So if I say, obviously there's the two divisions of the nervous system, which is the CNS and the PNS, but then they're organized by how do they control the body. There's either autonomic control or somatic. And if anybody has any questions about that, that's a very important thing to understand, is the control of the nervous system. Now, when you guys next term, you will take chemistry. It's not like chemistry like you're doing, don't think of it like experiments and the periodic table. It's almost kind of like biochem. So you're going to get into some of these concepts a little bit more in depth next term. But we have to talk about how does the signal get from the dendrite where you sense the signal And how does that signal make it all the way to the brain? So this concept's a little bit hard to understand, but there's something in the nervous system called the sodium potassium pump. And maybe those of you that are closely out of high school might remember talking about this in science or chemistry, or if you had a different degree. But what we're looking at on this picture, this is very important, so hopefully everyone can hear me. This is the inside of the nervous cell or the neuron, okay? So this blue side is the inside the cell. The yellow is the outside or the surrounding tissue outside the nervous cell. Remember, if you remember from the cell, I said the cell membrane had like this gate, okay? So this is the cell membrane. How things move in and out of the cell is controlled by this membrane, but there is, think of it like a doorway. And think of it, it's called the sodium potassium pump. Now, when in a normal, like you're chilling, sitting on your couch, nothing's happening, okay? You're in your resting state. On the inside of your cell, you'll see there's more potassium. See all those Ks? Ks with a little plus is potassium. If you go on the outside of the cell, you'll see there's more sodiums, which is the Na plus, okay? So when your body is at rest and the neuron's at rest, potassium is inside the cell, sodium's out. Now, if you look at the numbers, 1, 2, 3, 4, 5, 6, 7, 8. There's 8 potassium on the inside. There's about 9 sodium on the outside. So give or take 1. At rest, what I want you to know is, do you see how they're both positive? So you got positive, they are at equal numbers, roughly, okay? When they are equal, that creates what's called a net negative charge. If you look at resting membrane potential, they are negative, okay? So right now you have, because they cancel each other out, you have, if they're equal on both sides, the result is a negative charge. What I'm going to talk about is your nerve cells, when you're relaxing, are basically, they have the potential to fire and the potential to do something. So resting potential means that you have a net negative charge, you have equal particles on each side, and they basically cancel each other out and we get this negative resting. And that's the difference in the neuron membrane. Now think about an electrical charge. If it snaps in an electrical charge, I mean, that's like, ow. I mean, it's gonna excite or create something to happen. So what we're gonna look at here is what I always start off with, because I'm gonna ask you the order that it happens. It's like, what is the order once a neuron senses a signal, what happens next, okay? So when you're at resting state, the neuron is not being stimulated. Basically, sodium is outside the cell and potassium is inside the cell. And that equals a net negative charge. It's easier when I'm in the classroom, is I talk about you guys sitting in the classroom are potassium. We are the neuron. If I've got, there's 23 of you, if I've got 23 students in the hallway, you basically cancel each other out, okay? So right now, everybody's resting, everything's relaxed, sodium and potassium are equal. And what you need to know for the test is, I will ask you, like at resting state, is sodium on the outside or the inside of the cell? And you would tell me it's on the outside, okay? And think about something that's salty, and it's like, salting is excitatory. I'm gonna get to that. Okay, now, you start with resting state. The next thing that happens is, this is you touching the stove, or the dog cutting its paw, or something has happened to trigger that neuron, that nerve. So for some reason, your neuron you touch the stove, it now picks up a stimulus. There's heat. What's going to happen is you've got the outside of the cell and the inside of the cell is once this picks up a charge, the sodium gate opens and sodium rushes into the cell. So right now, sodium ions flow by passive diffusion. The charge in the cell is now positive. Think of it. When we were equal on both sides, we were negative charge. If I add a bunch of sodium to the cell, I add and I create a positive charge inside the cell. Do you see how I've added to the cell? So when sodium rushes in, if I ask you what phase of stimulus is sodium higher in the cell, you would tell me depolarization. Now, think about polar, what it means. You got the North Pole and the South Pole. You have polar opposites, okay? At resting state, you are polar equal. If I depolarize, I'm changing the polarity of the cell because sodium rushes in. So I've depolarized or changed the polarity because all that sodium rushes in. What that does is it creates something called an action potential. And that basically takes a cell from being at a negative charge to a positive charge. The way I think about this is now my neuron is excited and I have the potential to do something. The neuron has the potential now to send a signal. So this is what happens when a nerve is first stimulated. Sodium rushes in, okay? But what happens now, think about you guys in the classroom. Say like the fire alarm went off and all those 23 students rushed in my classroom. I've got 23 of you and I got 23 of them. I've just added students to my classroom, action potential. Well, now the cell is like, the cell always wants to make things equal, okay? The cells in your body like when things are equal on both sides. When all of that sodium rushes into the cell, you guys are like, okay, there's too many of us in here. We're going to leave. So what the cell wants to do is repolarize. But what happens in the beginning of repolarization is potassium flows out of the cell. What happens is 23 of you leave the classroom, but now I have 23 new students in the classroom and you guys left. Now I've repolarized. I've gone back to a negative charge, but I've changed positions. So at resting state, sodium is outside, potassium is inside. Nerve gets stimulated, sodium rushes in, I add or depolarize, and now my neuron is at a positive charge, which creates an action potential. After that happens, the cell is like, too many in here, but they switch positions. Now at repolarization, potassium is outside, sodium is inside. So hopefully that makes sense so far, okay? So at the beginning of repolarization, it's going to go back to a negative, it's going to start to go back to a negative charge because potassium is going to go outside to the point where they're both equal. And then when they're equal on both sides, that creates that net negative charge. Now, when we reach sufficient outflow of potassium, then we're going to get that negative charge. So right now, what you're trying to look at is, here is the inside, okay? But you see how they're all sodium. And then here's the outside, and they're all potassium. So when you have all that potassium flows out, you're going to get a negative charge inside the cell. Sodium and potassium are on the opposite sides. They've flipped. And then you're going to go back to a resting state. Now, what's going to happen? Okay. My computer is... Let me give it a chance to calm down here. Oh, let me see. When do they switch back to resting state with potassium on the inside? Okay, Savannah, that's a good question. What's going to happen? Everything I've explained to you, it happens at the very beginning of that stage. When you go back to resting state, well, when potassium is on the outside and sodium is on the inside, what's going to eventually happen is a few of the sodium are going to go back out and a few of the potassium are going to come back in. So as resting state gets longer, eventually they're going to go back to their normal sides. Does that answer your question? So it's at the very beginning. So at resting state that I started with, sodium out, potassium in, depolarization, sodium rushes in, repolarization, potassium rushes out, and then when enough potassium is on the outside, you do get a resting state again, but eventually a few are going to switch spots and you'll go back to the beginning of what I said. Does that answer your question, Savannah? Okay. And on a test, all I want you to basically tell me, or what I want you to know, is where sodium and potassium are at the very beginning stages of resting state, depolarization, repolarization. Obviously, after repolarization, you're going to go back to a resting state. But yeah, it's going to go. No, and then Savannah, that's a really good question because I kind of explained that in class. Like eventually it'll go back to that first resting state I explained where they'll start to kind of switch spots again. But the thing you'll talk about when we talk about a lot of things in the body is the body always wants things to be even on both sides. Okay, when we talk about oxygen movement, we talk about sodium, potassium, we talk about fluid molecules, protein. The body is always trying to make things equal on both sides. So that's how sodium, potassium move from themselves. Now let me see if I can get this to pull back up. Can you guys see this again now? Okay. Yeah, every once in a while, my little word just goes wonky. So I'm not going to get too much into this for this slide. So I just want you to understand the basic repolarization. But what happens, what I want to mention here is the nerve impulse. So I'm not going to get into the all or nothing principle, but nerve impulse is basically when the electrical charges flip. When you get an impulse, which is a pinched toe, you know, something happens, heat, pain, the electrical charges flip, and then what happens is you create a wave of depolarization. So let me get to that slide. So that's how an impulse moves from here to here, okay? We got to get that signal. Okay, so this slide number 21, I'm not going to get into refractory period. You don't need to know that. I want to get into what's called saltatory conduction. Think about saltatory, salt, sodium. So we're literally kind of talking about the movement of sodium. What happens here is, if you look over here, this would be the dendrite up here with the cell body, picked up the signal. Now it's coming down the axon. Basically, this cell here in this axon is going to depolarize. When this one depolarizes, it's basically going to create, this first cell goes into action potential. Then depolarization and axons takes place at the node of Ranvier. Once that signal gets to this node, it's going to trigger depolarization of this cell. It moves here, it's going to trigger depolarization of this cell. Basically, if I look at this nerve fiber, the signal cannot go from the beginning all the way to the end here. It's going to create this wave, and that's what's called saltatory conduction. It's basically this cell, the second cell, can't depolarize until the first cell does. So you get the first cell to depolarize, jumps, depolarizes, jumps, depolarizes. So it happens at that node of Ranvier. Then that signal moves the whole length of that axon, okay? And so each sodium channel opens, it stimulates the next sodium channel to open, then the next one, then the next one. So literally saltatory, salt, sodium, is the movement of sodium channels, and then it depolarizes all the way down the axon. Depolarizes, causes those that signal to be sent. Okay, now how they communicate to one another. I just talked about how the signal is sent. It's going to be easier if I look. Okay, there's 4 parts to kind of understand. A synapse is where a neuron joins or it connects to another neuron, or it connects to the target organ. That could be the intestine, the stomach, the bladder, wherever that neuron is controlling. When a neuron connects to either another neuron or an organ or another cell, the junction where they come together is called the synapse. So a synapse is that connection between the neuron and another effector cell or another neuron. Now, when they connect, there is technically a gap, and that is called the synaptic cleft. So that is the gap between 2 neurons. They look like they're connected, but there's a very little gap. There's a gap there. The presynaptic neuron is bringing the signal to that synapse, to that space, and then there's always a postsynaptic neuron ready to receive the transmitter, okay? If you remember, here's my axon. Here's my axon. Here's my dendrites. If you remember that tree, that neuron image, at the bottom were the roots, and I told you at the bottom of those roots were little telodendrons, and those little roots contained the neurotransmitter. This is closing up. This is a close-up image of one of the ends of those telodendrons. So that's what this picture is representing. So you see there's the axon, that's the tree trunk, this is the end, and this is the tip of just one of those roots that I said goes in the ground. So you have the telodendron. At the end of every telodendron is what's called the synaptic knob. It's like a little bump. So if we depolarize this neuron, that signal is coming down the axon, jumping through saltatory conduction. Once that impulse hits the end of this knob, it's going to cause the release of these neurotransmitters. So do you see how these neurotransmitters are stored in these little vesicles? So they're basically hanging out in there till an impulse comes down through the axon. So you have an axon, you have a telodendron. Here is a terminal bouton. Terminal is the end. So terminal is the very end or the tip. And then you have these vesicles. If you remember what a vesicle is, it's like a little package in a cell. So at the end, there's these little packages that are holding different neurotransmitters for different parts of the body. So those neurotransmitters are stored. Now, when you get a wave of depolarization, it's going to cause the vesicles to dump their neurotransmitters. So if you see these little circles, okay, these are the neurotransmitters that are releasing into the postsynaptic space. This is the presynaptic neuron. So this is what's bringing the signal. This is the postsynaptic membrane, this blue thing. This is either an effector cell. This could be a muscle. This could also look like another neuron. But what's happening is this is presynaptic space. This is postsynaptic space. This little gap is the synaptic cleft. So they are connected, but you see, it's almost like a moat. Think about like a moat around a castle. You've got like that water. It's kind of like a gap, and the neurotransmitter has to be released. But then, well, let me get to the next slide. When the neurotransmitter is released and they diffuse, they have to bind to a receptor on the postsynaptic membrane. So this is the presynaptic membrane, and they're going to bind to specific receptors. Neurotransmitters and receptors are like a lock and key. Or if you remember being a baby, there was that like circle toy, and you might have a triangle and a square and a circle, and only the circle goes in the circle, the square goes in the square. So basically, these neurotransmitters, when they're released, they can only bind if the postsynaptic membrane has its receptor. I'm going to give you an example. We're going to talk about a neurotransmitter called acetylcholine. Acetylcholine initiates muscle contraction in the somatic nervous system. So when you want to contract a muscle, the nervous system will release acetylcholine, and acetylcholine is only going to bind to receptors on the muscle cells, and that is what initiates a muscle contraction. So basically, acetylcholine, if it's floating, it's only going to have its action if that membrane has a receptor for it. I'll give you another example. When we talk about blood vessels, we're going to talk about fight and flight, and we're going to talk about stress. When we have a stressful event, blood pressure goes up. The reason it goes up is the nervous system releases epinephrine and norepinephrine from our adrenal gland. That norepinephrine binds to receptors on the blood vessels, and it causes vasoconstriction. So that neurotransmitter causes blood vessels to constrict. That increases blood pressure, which helps increase blood flow, because the body in fight and flight or stress is preparing. They think you have to fight. I mean, it could be somebody jumping around the corner and scaring you. It could be obviously dogs in a dog fight, or a hit by car, or anything that causes that reaction. We get a release of norepinephrine, epinephrine, and then it binds to blood vessels. But norepinephrine and epinephrine aren't going to go to every cell in the body and cause a response. It has specific job and a specific receptor. So obviously I'm going to talk about that when we get a little bit further, but what I want you to understand is that you have a presynaptic space, you have a postsynaptic membrane, you have a synaptic cleft, and this whole thing is called the synapse. This is where we connect together, okay? Now, I was just talking about the types of neurotransmitters that we have. Now, when we need our nerves to do something, we need what's called excitatory neurotransmitters. And excitatory neurotransmitters usually cause the postsynaptic membrane to influx sodium, which excites or it increases a threshold, and it helps create a new impulse. So most excitatory transmitters obviously cause something to happen, excitatory depolarization. On the flip side, whenever there's an excitement, we're going to have to calm down or inhibit a response. Inhibitory neurotransmitters are going to make the cell more negative, and they're going to kind of prevent that postsynaptic membrane from exciting, and then we move the impulse away and we don't cause a response. Now, if you've ever like went to touch something and you're kind of like, oh, it's not hot, okay? So you didn't go like this, you kind of just went, oh, okay. Do you see how that kind of like calmed down? So sometimes the body's going to need to really excite something, and there's excitatory, and then we're going to have to calm it down. Now, the ones that I want you to know, this is slide 28. You need to know acetylcholine, and then we need to know the catecholamines. The catecholamines are the norepinephrine, epinephrine, and dopamine. Those are all excitatory. GABA is the only one I want you to know that's inhibitory. For right now, I'm just going to talk about those. So acetylcholine is a very important neurotransmitter. It helps control muscle contraction. Acetylcholine also affects the parasympathetic, or which is the, hopefully you guys, a lot of people have heard about sympathetic and parasympathetic, which is sympathetic is fight and flight, parasympathetic is rest and restore. Acetylcholine is also a parasympathetic neurotransmitter. So acetylcholine has multiple jobs. It helps our somatic nervous system, so it helps your muscle contraction, it's the primary neurotransmitter, but it also calms things down. So acetylcholine really has two jobs. Catecholamines, norepinephrine and epinephrine, are obviously excitatory. And what I will tell you, and I know some of you have experience and have maybe been in a clinic, but most people that even watch Grey's Anatomy, and they say push epi, okay? Epinephrine we have in drug form, and it's one of the primary drugs we use in cardiac arrest. If you have an animal that needs CPR, we typically give atropine, and we typically give epinephrine. Sometimes it depends on the type of cardiac arrest, but that's probably the first two drugs that we are going to look for. So a lot of these neurotransmitters, these that I'm talking about are natural in our body, but we have epinephrine. We have a drug called phenylephrine that kind of can bring up blood pressure. So some of these drugs, and if you've ever heard of gabapentin in a clinic, usually gabapentin is used for like neuropathic pain. And what it does is it calms the nerves. So your nerves aren't firing in pain. It's going to try to suppress that. So hopefully this will start to make sense when you get into pharmacology, or you're in a practice and you're prescribing GABA, or you're using epinephrine for cardiac arrest. A lot of these chemicals are natural in the body. Sometimes we use them pharmacologically to mimic and help the body do what we want it to do, okay? So these are the primary neurotransmitters that we'll talk about. And then now the body likes to recycle things. It's kind of like a big recycling plant. When the body releases acetylcholine, and acetylcholine is doing its job, if the body wants to stop the effect of acetylcholine, yes, I can do that one. I'll do that 1 second. If it wants to stop acetylcholine, it'll release something called acetylcholinesterase. A lot of these, you can see they all have ACE, esterase, oxidase, erasing the effects. So that's the way I think of it. Acetylcholinesterase is going to erase or stop the effects of acetylcholine. Monoamine oxidase is going to work more with the excitatory neurotransmitters like the catecholamine, with the dopamine. And then you've got catechol. That's going to stop the acetylcholine, or the catecholamines. So let me type that out really quick so that you know. Let's see. Okay, so I typed out acetylcholine. It's just, you'll hear a lot more about acetylcholine. We don't really talk about epinephrine and norepinephrine unless we're talking about the sympathetic system, which is your fight and flight. So I get into those a little bit more when I do the endocrine system because they are produced by the adrenal gland and they're the primary neurotransmitters of stress. So when there is a stress response, you need to have functioning epinephrine and norepinephrine because that helps raise your blood pressure, which supports your heart rate, which gets more blood flow to your muscles because the body's like preparing you to run. It helps open up your airways so you can take in more air. So there's like a lot of effects that we're going to get into when I get into the sympathetic and parasympathetic system. And that will make more sense with these neurotransmitters. I'm assuming you guys have math at 11. Yeah. I think I'm going to stop here because we're going to start to get into some of the parts. We're going to talk about the parts of the brain. I'm going to send another email because I have a few links that I want to send you. It's going to take me some time because I want to go through some different videos. I had some of them saved at my work computer, so I need to kind of dig through my old stuff and see if I can find these links. But we're going to talk about like cerebellar hypoplasia, for example. So we're going to talk about the parts of the brain, what do they control, and then why you see the diseases. If the disease affects that part of the brain, why does the animal act that way? So we're going to get into this section next. Hopefully if you bopped on here late, I sent you through I opened the assignment through Teacheries for you to submit your homework. I did not send I didn't link the document to Teacheries. I know Zuber told me I could do it either way, where I email you the document and then you just upload it into Teacheries. So I did not link the document to that assignment because she said then that kind of messes things up. So does everybody understand? There's the homework sheet, exercise 13 in the nervous system lab book, just one exercise. And I just want to have that due Tuesday. You don't have school tomorrow. So maybe you can just break up these. Yes, both will be to Tuesday. The homework sheet and the lab exercise. If you want to just write out, the lab exercise is literally terms. If you want to just type those out in a Word document, if you want to write them in, I mean, I guess you can upload it. If you can't upload your picture, you could always email me, separately at my school email, whatever is easier for you. Okay, let me unmute. I've unmuted everybody. Does anyone have any questions?
No, I don't have any questions. This is Sean.
Okay, hi Sean. If I'm trying to get, hang on a second.